Alright, so today I'm going to be giving the solutions to the annotation worksheet, the first one. In case you haven't had molecular biology, or at least enough biology to kind of get by in annotation, sometimes it can be confusing coming in with some of the concepts. You may not be as familiar as you want to be. I know for me I had to do some brushing up as well. So I designed this worksheet to just make sure that certain concepts were solidified to have a good foundation for going into annotation. So I'll just begin. The order of the questions might be different, or maybe there might be like a new question. As I was putting in the solutions, I felt like it flowed better in different ways, so I made slight adjustments, but it should be about the same. The central dogma of molecular biology is essentially that genetic information is in the form of DNA, and through the process of transcription, the DNA is turned into RNA, and we'll get more into transcription in a bit. The difference between DNA and RNA is that the RNA has a uracil instead of a thymine and is single-stranded and uses a ribose sugar instead of a deoxyribose. So those are the main differences, but the coding is about the same. This is happening in the nucleus for eukaryotes. After transcription happens, the mRNA transcript is exported to the cytoplasm, where it can interact with ribosomes to get translated into a protein. And you start with the start codon that makes methionine, and it keeps going until it hits a stop codon. A lot of the words I just used we'll re-see again later on. So the process of transcription. So if this is the form of the question you got, I might reword this to specifically say, don't worry about the differences between prokaryotes and eukaryotes as much, because there is a difference. Transcription, just to keep it basic, is like I described in the central dogma. It's this step here, going from DNA to RNA. That's it at its fundamental core, and I think if you just understand that, you're fine. But if you are curious about a little bit more detail, not to get too pedantic, but it's broken down into 3 steps, just like translation is. And it might be a little confusing because the steps for transcription and translation, the 3 of them are the same. It's initiation, elongation, and termination for both processes. But in initiation, you have an enzyme called RNA polymerase that's going to bind to a promoter region. In your DNA, there's going to be like a sequence of DNA called a promoter, and that's going to recruit the DNA polymerase, and then maybe some other enzymes as well, but not to get into that detail. But it's just upstream, or 5' if we're talking directionality, of your open reading frame for your gene. After the RNA polymerase binds, the 2 DNA strands will separate of your double helix, and this RNA polymerase enzyme will start to build the RNA chain. So it's adding on nucleotides through, I think, the beta subunit of RNA polymerase. After these nucleotides are added on, it's essentially ready to start elongating. It puts on the initial few, then something called promoter clearance happens. It sort of shifts. The subunits of the enzyme sort of dissociate, and only particular ones stay on. And then the mRNA transcript continues to get elongated. So the RNA polymerase keeps synthesizing on in the 5' to 3' direction. To pay attention to directionality, the polymerase enzyme is reading, if this strand here is going 5' to 3', the RNA polymerase is reading the DNA in the 3' to 5' direction, but is synthesizing an mRNA transcript in the 5' to 3' direction because it's complementary. This strand would be your template strand because it's serving as the template for which the mRNA transcript is based on. And the opposite strand is known as the coding strand. The reason that is because when your mRNA is synthesized, it's going, even though it's using this strand in the 3' to 5' direction as the template, the sequence is going to match up with that complementary strand, so on the opposite strand. So actually, to skip a little bit ahead, here you'll see a little bit better picture. 3' to 5' direction, that is your template strand. And the RNA polymerase is acting on here, and it's synthesizing in the 5' to 3' direction. But the mRNA here is the same sequence as the coding strand, even though it's not the parent. It has the same sequence, except, of course, the thymines are switched out with uracils. Then termination. So termination, you don't need to know too much about it, but there is a terminator sequence, and I think this may vary. Yeah, this varies a little bit in prokaryotes versus eukaryotes. Don't worry too much about it. But there is a terminator region that kind of signals for the end of transcription. The RNA and DNA become dissociated from each other, and the transcription will stop. So in prokaryotes, there is an enzyme that sometimes pops up called Rho that assists in this, but don't worry too much about that. So just to keep it basic, you have your initiation, elongation, and termination, and just generally knowing that this happens during it. Not worrying about the specifics of the differences between prokaryotes and eukaryotes is fine. Though, of course, with D-Citry genome annotation, we are, of course, working with a eukaryotic organism. So if you did want to look into the specifics of how eukaryotes transcribe, and of course, it's more complicated than a prokaryote, just because eukaryotes are more complicated organisms, you're welcome to. I refrain from putting any notes on it in here. But I did put this little chart. You're welcome to look through it, but it basically goes in and compares prokaryotic and eukaryotic transcription just to show that there are differences in them. It's not super necessary. It's just a little fun fact. As we already discussed, DNA is transcribed into RNA in the 5' to 3' direction, so the mRNA transcript is getting added on the 3' end in the 5' to 3' direction. But of course, the DNA on the template strand is 3' to 5'. So what does it mean when I say sense or coding versus anti-sense or non-coding? Sense, aka coding, that's the opposite of the template. I should probably put template, but like anti-sense, non-coding, template, they're all synonymous. I kind of hate that there's 3 terms for each of these, and sometimes it's very easy to get them mixed up, but just in case you see these terms and then template, my favorite pairing is coding and non-coding, or a template and coding. Those are kind of easy. The template is pretty intuitive because it's the template DNA for the RNA, and then the coding it has the same code. So as I kind of already went over this, but the 3 differences between DNA and RNA is, of course, the uracil instead of thymine. Did I spell that right? There we go. Single versus double-stranded, and then here's a little picture. I'll make it a bit bigger. There's a ribose sugar instead of a deoxyribose. So this deoxy, if it didn't have the deoxy, it would just be ribose. But what that means is here, if you look at the RNA, there are 2 hydroxyl groups. That's what you have, like an OH. So that would make like an alcohol if you've done organic chemistry. But these hydroxyl groups, the OHs, they're generally pretty reactive, I think, because they're nucleophilic. It's been forever since organic, so forgive me. But then the deoxy, so it's lacking oxygen, and that's why it's named this, because there's an extra oxygen on the ribose versus the deoxy. If you're familiar with chain termination sequencing, then you can have dideoxy, where you lose both of the hydroxyl groups, which is interesting. Just to keep it basic, yeah, this is the main differences between DNA and RNA, just good to review. Don't worry about the structures of any of these, it's not important. But all right, introns versus exons. So this is good to know. This is kind of like something having a strong understanding of is beneficial because you work with this literally all the time. So this is specific to eukaryotic cells, and it would be helpful if I had Apollo pulled up, but I don't. But eukaryotic cells have this thing called mRNA processing, which I get to in the next question. But essentially, DNA has a lot of non-coding information in it. There's long stretches where the DNA exists, and it's not that it's not important, because introns are important, and I'll get into that in processing as to why they're important. They do not, after the splicing occurs, the introns do not contribute to coding a sequence of an amino acid. So they don't get translated, but they do have a function, and I'll get to that. Introns and exons, this is only happening in eukaryotic cells. Prokaryotic cells are simple, and they don't have all that non-coding material. So there's here called hRNA. There's another question where I ask what the difference between hRNA, cDNA, and mRNA are. But hnRNA is the sort of raw product as transcription is occurring. As the RNA transcript is being made initially, it's called hnRNA when it's still unprocessed. When mRNA processing and splicing occurs, then it turns into regular mRNA transcript. So introns are spliced out by enzymes called SNRPs, and the exons are the coding regions that stay, that eventually get turned into amino acids. So introns are important because they have sequences inside of them that help with splicing. So like I said before, they are important. And actually mutations in them can lead to splicing. I had a project once where I actually looked at a disease, I think called Fabry's disease, and if you want to look that up, you're welcome to. In an intronic region, there was a mutation that got rid of a splicing signal, and so incorrect splicing occurred, and that's what actually caused the mutation. So just because it's not coding doesn't mean it's not important. And here, you can see a little bit more about what an intron structure would look like. So there's the 5', here would be your first and your second exon, just as an example. If you have like a long gene, maybe, it's common to come across genes that have like 20 or even hundreds of exons. And, you've seen gene models in Apollo, but in Apollo, you just kind of see these straight black lines, and it's very easy to kind of forget how important they actually are. Here in this intron, you have a 5' splice site, so there's like a sequence, like a consensus sequence. Here, there's the branch point sequence, then there's this 3' splice site. And this branch point sequence is actually extremely important, and there's a particular A, so an adenine in this branch point sequence. It's called the central A, or the branch point A. Whenever your SNRPs come in, and here's a little bit of a diagram of how the SNRPs work, but U1, U2, U5, 6, there's like 6 SNRPs. These are all like the subunits of them. So they come in a particular order. Don't worry about the order. But the SNRPs come in. They recognize the branch point A and the splice sites on either side. By recognizing these signals, they come together and they kind of like cut them apart and then paste the exons together, and the introns leave. Sometimes the introns just leave as a straight line. Oftentimes you also see this lariat structure forms, which is when the 5' splice site will actually attach on to the branch point A, or vice versa, I forget. And then it forms this little loop-de-loop. Then the 5' splice site then attacks the 3' splice site, and then the lariat gets broken off, and then the exons form together. So yes, I recognize this is a little bit detailed and pedantic. You don't need to worry too much about it, but just to give you a little bit of excess information as to how mRNA processing works, you're essentially taking your hnRNA, enzymes act on it, introns are removed, exons are joined together. So then you're left with just your spliced mRNA. So now talking about UTR. So in molecular biology, there was like a whole chapter dedicated to the 5' cap and a whole chapter dedicated to the poly A tail. I just took my basic notes from them. But essentially, the cap and tail function similarly. You don't have to worry about cap structure so much, but this is what it does, essentially. There's an enzyme called RNase, which anytime you have ACE at the end, that means that it degrades. By having this cap, it's going to defend the mRNA transcript from being degraded by this enzyme. And this enzyme, RNase, is just kind of present everywhere. So having some sort of way to defend it and protect it from being degraded is nice. It increases translatability, so whenever the mRNA gets to the cytoplasm and interacts with the ribosome, the 5' cap is going to help the transcript be recognized by the ribosome, and it's going to start help with that first round of translation, and I'll get into translation later. It aids in the transport out of the nucleus. In the first intron, or the first exon, in this, where we're talking about the first intron, where it's the splicing between the first and second exons, the 5' cap, which would be here, not depicted on the left, would actually assist in this process happening here. So kind of multifunctional, a little bit detailed. Next we have the 3' cap, or the 3' UTR, and oftentimes it'll have this poly A tail, I think always actually, but the A stands for adenine, so it's that one nucleotide, but it's usually around 250 adenines long, though it can vary, I think, a little bit. But again, it's going to protect from degradation from RNase, just like the cap does. Essentially, by having these 250 adenines on at the end, the RNase acts on the endpoints first. So by having these caps on the end, it's like the important information is not getting harmed in the middle, instead it's these caps. Same as it increases translatability. The poly A tail helps with association with the ribosome. helps with splicing in the exact same way as the cap does, but just on the 3' end instead of the 5' end. Don't worry about this one too much. This is like a whole process, but essentially the poly 8 circularizes, and by being a circle, it's more stable than it being linear RNA, because then the RNase can't easily access the ends to form a cap. I think there's like a helper enzyme that sort of like binds them together. I forget the details, but cDNA, or complementary DNA, is often in nature found in the case of retroviruses. And retroviruses have a single-stranded RNA genome rather than a double-stranded DNA genome, which is what eukaryotes have. So since they have an RNA genome, often to sort of integrate into the hosts and hijack their host machinery, they use an enzyme called reverse transcriptase to change the RNA into DNA rather than DNA into RNA with RNA polymerase. So this is kind of going in the reverse direction of the first step of the central dogma, where instead of DNA to RNA with RNA polymerase, you're getting RNA to DNA with reverse transcriptase. Regular DNA polymerase, just like when DNA replication would happen, you'll get complementary DNA. So that is cDNA. It's after mRNA. It's like converting mRNA into DNA. I'm trying to think of this, if there's any. I don't want to get into like cDNA libraries or anything like that. I will say the reason why in lab we tend to prefer working with cDNA is because, kind of like has been alluded to, mRNA is not stable. The reason is that RNase enzymes are just everywhere all the time. So RNA will get degraded very easily, and DNA is a lot more stable. So kind of working with cDNA, you're going to have a lot less risk of your RNA getting degraded by RNase. Alternative splicing. So we talked about splicing already. So the SNRPs come in, the introns are kind of helping out where they get spliced out. But it's not only the introns that'll get spliced out. Sometimes exons get spliced out too. So you have your pre-mRNA, also called hnRNA, or heterologous nuclear RNA. And let's say, for example, you have 4 exons here. So you have your red, yellow, green, and blue. Let's say that here we want to splice out this green exon. So instead of just the introns getting removed, the whole entire green exon in addition to the introns will get taken out. So then you'll have this version of the mRNA transcript where it's missing one of the exons. But then let's say that in the intron sequence there's kind of like some ambiguity, or like an alternative code, like a different way it could be reinterpreted, where sometimes, depending on what like other outside factors or enzymes or proteins are working, it will splice differently, and that's called an alternative splice site. These exist in the introns, like signals in the introns, which is another reason why the introns are important. And gene expression will happen, and a certain type of the protein will be required, but at another instance, maybe a different form of the protein is required. So from the same gene, by selectively choosing which exons to include, we get functionally 2 different proteins, similar but different proteins. Here we have a red, yellow, blue exon. So we have exons 1, 2, and 4, and here we have 1, 3, and 4. So you actually get a different isoform of a protein. And you can actually see this in Apollo. I've actually come across an alternative splicing site where I had to create isoforms to account for in a gene model. And oftentimes you'll see this as RNA-seq having kind of an ambiguous, like it looks like there's 2 different ways that it could go. And I think in the 3rd worksheet with Apollo, I think I actually have a question where I ask you that. If not, I'll add it back in. And why do we do this, right? So essentially what's happening is we're getting 2 different proteins, or at least 2 different protein isoforms, from a single gene. The gene makes the RNA, and the RNA can then make 2 different proteins, or even more than that, right? Like, this isn't limited to just 2. This is just with an example of 4 exons. But in a gene where you have hundreds of exons, the possibilities are endless, really. Well, not endless, but considerable. And so why is this nice? Well, the genome is only so big, but the proteome is much larger than the genome. And we talk about genomes and proteomes later. So ORF. The open reading frame is just it's starting at the start codon, and it's going and stopping right before the stop codon. So an open reading frame, like if you have your promoter upstream 5' of your start codon, and then you have your AUG methionine, will be the first amino acid, and then it'll keep reading until you hit a stop codon, TAG. So everything that's in that reading frame that is translated into a protein, that is considered the open reading frame. But the thing is, with a strand of DNA, they're grouped into codons, right? It's 3 nucleotides at a time. The thing is, you don't know what the first letter is, right? You don't know what the first letter in the codon is. So here are 3 of the exact same sequences, right? So in this first one, we have it where the first letter is the start of the first codon. So you have ATG, which translates to AUG, which is your start codon. In the second reading frame, however, we don't start at the A, we start at the T. And this changes every single codon. We call this a frame shift. Like, instead of starting at T, what if the A just got deleted? Then the reading frame would be completely shifted, and instead of reading properly like the first frame, we instead would get it reading like the second frame. This, in this first frame, A is the first letter, then T is the first letter, and then G is the first letter. And this changes what the open reading frame is. So in frame 1, we get this full sequence, but in frame 2, we don't get a start codon until way later, and then we get a stop codon way earlier. So you get this truncated protein. And then in frame 3, we have the exact same thing happening, where it's starting way later, it's getting truncated in compared to the first frame. A single nucleotide change can potentially frame shift and just wreck your whole protein, so you can get this second one instead of a nice complete first one. All right. Okay, so then particularly the kind of tricky part I asked are, how many possible reading frames are there for a double-stranded piece of DNA where it is unclear which strand is the sense and which is the antisense strand? So what I mean by this is, if you have your double-stranded DNA here, so let's say this will be 1, 2, and 3. This will be 1, 2, and 3. So you have 2 strands of DNA. You don't know which one is the template strand. There are actually 6 different reading frames that could be here, because you don't know which direction it's facing. And in Apollo, you'll actually see this, especially in the third worksheet. There's a particular question that has you interpret in Apollo how the open reading frame is. In this direction, you have 1, 2, and 3 different open reading frames, as we just discussed. But if we flip this around, it's kind of trippy, it can be read also in this way. This is 3' to 5', like how you would interpret mRNA. So you would get your RNA transcript being read off this way. So instead of properly how you have your coding strand, your coding strand would become your template strand. So there's actually 6 different open reading frames in a double-stranded piece of DNA. And sometimes in Biotech 2 lab, there was actually an instance where Dr. D'Elia gave a sequence of DNA, and he asked us to figure out what the open reading frame was of the 6 combinations. So we actually used a program called MBOSS to generate a protein translate, like a electric translated protein, from each possible open reading frame. And then we had to blast that protein and see if it came up as positive. And the one protein that was blasting correctly, we knew to be the correct open reading frame. So you can actually just determine that through using MBOSS and BLAST, which is pretty cool. Don't worry about MBOSS. I don't think I've ever used it for annotation. It was more of a just a general biotech thing. All right, so translation now. So we pretty much beat transcription like a dead horse. I feel like we've gone through it decently. But now translation. So I included this here YouTube link that goes through a little animation of how it works. Just to start off, I wanted to talk a little bit about pre-initiation. Translation as a whole is just, in one sentence, changing from mRNA into protein, right? And the specifics of that process are the mRNA transcript goes to the cytoplasm and interacts with the ribosomes. So how this happens is we have things called tRNAs, which are these, they kind of look like T's, and they have I wish I had pulled up a picture, but all right, I pulled up a picture of a tRNA, and what this tRNA does is it has this anticodon, which has a complementary sequence to whatever your codon is. And this tRNA, called a transfer RNA, has the amino acid that's going to be added onto the polypeptide attached to its 3' end. So an enzyme called aminoacyl tRNA synthetase attaches on the amino acid to this 3' tail of the tRNA. When we get into the translation process in initiation and elongation, there's going to be peptidyltransferase activity that is actually a part of the ribosome, and this is when the amino acids are sort of attached to each other, and this will make a little bit more sense. I just wanted to put these terms up front because I didn't want to have to define them down here. But in pre-initiation, you have tRNA charging. So this is when you have your tRNAs that kind of just float around, and when they're needed, the enzyme aminoacyl tRNA synthetase will attach the amino acid here, and that's important in pre-initiation. And then the ribosomal subunits will dissociate. Oftentimes, think of these ribosomes as factories. They're constantly churning out proteins, churning out proteins. So at the end of a cycle of the protein being created of the last one, you need to have the big and small subunits dissociate so that you can start the process over again. So after dissociation, then translation can start. So kind of pedantically, pre-initiation is not considered a part of translation. That was actually a test question in molecular biology. It is important. It happens as a prerequisite, but it's not considered part of the process. The process is comprised of initiation, elongation, and termination. And I kept it really bare bones here because you can get kind of in depth into it. I put the YouTube video here that kind of shows an animation of how it happens, but don't worry about it too much. But yeah, so in initiation, you have your mRNA transcript binding to the ribosome, and because of what we went through before, you know that the 5' cap assists in the mRNA binding and associating with the ribosome. So then the first charged tRNA, which has which amino acid? That's methionine. It binds to the first start codon, which should be AUG. And then all of your initiation factors bind, and there's a whole plethora of them for eukaryotes, and they're actually different in prokaryotes. So then elongation happens, and this is where this diagram with the APE site, and this video I think more so covers specifically elongation, where the let's say in the first round, tRNA with your methionine comes into the A, and then it shifts to the P. So now there's a tRNA with a methionine here in the P site. Then a new tRNA will come in. It will go into the A site. Peptidyltransferase activity from the ribosome will bind the amino acids together from the P to the A site. And then the amino acid chain, the polypeptide chain that's growing, will be attached to the tRNA in the A site. Then everything will translocate one round. What was the tRNA that was in the P site no longer is holding on to the amino acid chain, and it shifts to the E site as the A site shifts to the P site. At the E, which is the exit site, the uncharged tRNA will dip, and now we have what was in the A site becomes into the P site. That probably, it doesn't make a lot of sense if I'm just saying big words, but watching the animation is helpful, so I'd recommend that. All right, so that's elongation, and then termination. So I will just cover this in this next question because it talks about termination. To keep it basic, essentially, and this differs between eukaryotes and prokaryotes a little bit. Essentially, the important thing I wanted you to know was that there is a stop codon. So there's three different stop codons: AUG, UGA, and UAA, and release factors recognize these stop codons, and release factors are kind of shaped like tRNA, so they kind of fit into like these APE sites easily. And the release factor will bind. It kind of mimics a tRNA, but it's not. A process occurs and translation ends. Yeah, I won't go into more than that. But here's some details on the release factors. Wobble position. I talked a bit about in person the specificity of cDNA, and I think there might be a question on it actually. Yeah, there is this next question. Yeah, so these two questions kind of relate, but essentially two peptide sequences can be equal, but their mRNA or cDNA sequence can differ. And the reason is normally nucleotides obey Chargaff's rule, which is the base pairing rules between nucleotides. So you get normal GC UA pairing, and that's called Chargaff pairing. But it doesn't always have to be the case. In the third position of your codon, the third letter, sometimes you can have improper pairing. So normally G always binds with C, but we can change it so that G binds with U instead. This just happens and it's fine. Just because the code changed, you still get the same amino acid. Kind of demonstrating that in a little bit more of an applied setting. I asked to provide an amino acid sequence with these specifications and also two cDNA sequences, so I did that here. For the amino acid, I chose methionine, tyrosine, cysteine, and then the stop codon. So the methionine is always going to be UAG. Here you can see this codon usage chart. AUG will always be methionine. That's the only option for it. UAU, however, let me find that. That was tyrosine. Tyrosine's here. UAU or UAC. There could be this wobble pairing in the third position, and you'll still get tyrosine. And you can kind of see that illustrated here. Same with cysteine. So UGU, or you could have that last position wobble paired, and you can have UGC, and you'll still get cysteine. So even though these two mRNA sequences are different, the amino acid sequence is the same. And when we're talking about specificity, maybe it's a little bit confusing because I use the 3-letter code for amino acids. I don't remember the I don't have all the 1-letter codes memorized. But this is just one amino acid, one amino acid, one amino acid. So there's only 3 amino acids here, 3 residues. However, there are 9 nucleotides in this one. So it's 3 times as much. I didn't include the stop codon, actually. That should be there. There we go. Yeah. So including the stop codon, it's 12. So you're getting 12 nucleotides for 3 amino acids. So your mRNA is always going to be longer and also more specific because of this wobble pairing. You can have different mRNA codes, but of the same amino acid. So this is why when you're in Apollo, using cDNA pairwise blasting or cDNA blats will give you a more specific hit. So comparing contrasts, residue, amino acid, polypeptide, and protein. This is pretty simple. Residue is the same thing as amino acid, and you'll actually see this a lot in annotation. You'll see things referred to as residue, so knowing these are the same is helpful for your brain. Polypeptide is when there is a string of amino acids. So think of the amino acids as the building blocks. Polypeptide is more than one peptide. And then a protein is when you have a complete polypeptide. Polypeptides can be incomplete. They don't have to, there's no rule as long as there's multiple of them in a row, that's a polypeptide. But a protein is specifically like a folded functional, unless it's denatured. But a protein is just a folded polypeptide that has a certain structure. And just to briefly go over protein folding, you have your polypeptide here, and then that's considered the primary structure, and then it starts to form these secondary structures by interacting in certain ways because of the charges and properties of their residues, or their side chains. And so you can either get alpha helixes or beta pleats or sheets. Then as they start to form more complex shapes, you'll get a tertiary structure, which is can be like a domain or something. And then the quaternary structure is when there's multiple tertiary structures folded together. So that's from least complex to most complex, and that is protein folding. So that kind of separates the peptides from the polypeptides from the proteins. So this is obvious by now. I've gone through this many times, but the first amino acid in a polypeptide is methionine, and in Apollo you will see methionine. Making sure that methionine is the very first thing you see at the start of a gene model is important for validating and annotating. So what is gene expression? Genes exist in the genome, just they exist. However, they're not always active. When a gene is expressed, that means that it eventually makes a protein. So gene expression refers to when a gene is turned on, which causes transcription and translation to occur, and then a protein is made. So it's that when the gene is turned on and a protein is made because of it. And then the last question is compare and contrast a genome and proteome. So mainly I included this question because you'll see the word genome a lot, just to have a solid understanding of what a genome is. It is the entire genetic material of an organism. So all of the DNA, including the non-coding stuff, so like your introns and stuff, it's just all the genetic material, and that includes your mitochondrial DNA, or if you're a plant, chloroplast DNA, because as you know, hopefully, mitochondria and chloroplasts have DNA. That can be explained by the endosymbiotic theory, if you're familiar with it, which is the fact that mitochondria and chloroplasts used to be prokaryotic single-cell organisms that mutually sort of adapted, evolved, and became a part of us. Well, we don't have chloroplasts, but I digress. So that is the genome. And the proteome is the entire set of proteins that can be expressed, again, gene expression, by a genome. Yeah, so that's including all of the alternative spliced proteins and everything like that. So just so you know. So that is the entirety of it. Thank you for watching, and I hope that by doing this worksheet it was helpful. Have a good day, and I'll see you next time.